Assalamu alaikum. I am Dr. Ehsanul, Associate Professor of Biochemistry at the Akij Medical College, Khulna. Today our topic of discussion is digestion and absorption of carbohydrate. So carbohydrate is a chemical substance composed of carbon, hydrogen and oxygen. And this is probably the most abundant chemical substance in nature, and it is a very integral part of our biological system. It is the major source of energy and it helps in various physiological function in our body. So what are the dietary sources of carbohydrates or dietary forms of carbohydrate? The main dietary forms of carbohydrates are starch, lactose, sucrose and cellulose. These are basically disaccharide and polysaccharides which are formed by the connecting the monosaccharides by glycosidic bond. The principal site of carbohydrate digestions are mouth cavity and intestinal lumen. The digestion of carbohydrate is very rapid and it is done by the enzyme glycosidases or glycoside hydrolases which hydrolyze the glycosidic bond. And after the breakdown of glycosidic bond, the final products of carbohydrate digestions contains glucose, fructose and galactose. And these are ultimately absorbed by the intestinal cells. So let's talk about the digestion process inside our mouth. In our mouth, the digestive juice available is saliva, and saliva contains an enzyme called salivary alpha amylase. The salivary alpha amylase acts upon starch, which is the major dietary polysaccharide from plant. And this starch contains amylose and amylopectin. Salivary alpha amylase also acts upon the glycogen, which is the animal source of carbohydrate. During mastication, salivary alpha amylase briefly acts on starch and glycogen and breaks down the alpha-1,4 glycosidic bond. But amylopectin and glycogen also contain 1,6 alpha glycosidic bond, which the salivary alpha amylase cannot break. So the digestion of carbohydrate inside our mouth is fairly partial. It is not a complete digestion, as the 1,6 glycosidic bond is not broken down by this salivary alpha amylase. After the brief digestion process inside our mouth cavity, the digest contains a mixture of short, branch and unbranched oligosaccharide, which is called dextrin. And there is also isomaltase, maltose and maltotriose available in the digest. But the disaccharides, that is sucrose and lactose, are not digested. Also cellulose, which is a polysaccharide, is not digested inside our mouth because there is no enzyme available inside our mouth to digest those. So after the mouth, the food content enters into the stomach. But inside the stomach, the digestion halts temporarily because of the low pH or the high acidity of the stomach content, and the activity of salivary alpha amylase temporarily halts there. So after the stomach, food content of the stomach enters into the small intestine. In the small intestine, the pancreatic juice containing bicarbonate neutralize the acidic content of the food. And also the salivary alpha amylase also the pancreatic alpha amylase, which is available in the pancreatic juice, starts their action and continues the carbohydrate digestion. The final digestion process takes place primarily at the mucosal lining of the upper jejunum. Here disaccharides like isomaltase, maltase, sucrase, trehalase starts their action. Isomaltase breaks down the alpha-1,6 glycosidic bond of isomaltose. Maltase breaks down the 1,4 glycosidic bond of maltose and maltotriose, and each producing glucose. Sucrase breaks down the 1,2 alpha glycosidic bond of sucrose producing glucose and fructose. And lactase breaks down the beta-1,4 glycosidic bond of lactose producing glucose and galactose. So after the digestion process, the end product we get from carbohydrates are glucose, galactose and fructose. So all these end products of carbohydrate has different types of absorption system or mechanism, and they are ultimately absorbed inside the enterocyte. For example, glucose and galactose are transported into the enterocyte by a secondary active transport system called sodium-dependent glucose cotransporter 1. It is a secondary active transport process, and it uses a concurrent uptake of sodium ions. So, fructose is another end product of carbohydrate. Fructose utilizes glucose transporter 5, GLUT5 we say, for its transport across the enterocyte. So all the monosaccharides, glucose, galactose and fructose are transported from the cell to the portal circulation by yet another transporter called GLUT2. So this is how the end products of carbohydrate enters into the circulation. Now let's talk about some abnormalities of carbohydrate digestion. The carbohydrate digestion in normal human body is so efficient that by the time the food content enters into the upper jejunum, almost all the carbohydrate substance are digested and absorbed nicely. However, because only monosaccharides can be absorbed, if there is a deficiency in disaccharidase enzymes, there will be passage of undigested carbohydrate into the large intestine, and it will cause osmotic diarrhea because these substances are osmotically active. This will ultimately cause abdominal cramp, diarrhea and flatulence because these are sometimes fermented by the intestinal bacteria. Another important abnormalities of carbohydrate digestion is lactose intolerance. In fact, most of the adult population, there is more than 70% of the adult population in the world are lactose intolerant, and some ethnic population are more intolerant than other, like the Asians and Africans descents are more than 90% people are lactose intolerant. So the treatment of lactose intolerance is basically avoiding the milk product and use alternate substance for the nutrition. Another abnormality is congenital sucrase isomaltase deficiency, and this is actually an autosomal recessive disorder which results in intolerance of ingested sucrase. So how can we diagnose these abnormalities of disaccharides? We can diagnose these enzymes deficiency easily by tolerance tests, oral tolerance tests of this specific disaccharides. Also, the measurement of hydrogen gas in the blood is a very reliable test for the determining of the amount of ingested carbohydrate which is not absorbed by the body but rather absorbed by the fermented by the bacteria of the intestine. So this is all about the digestion and absorption of carbohydrate and some of the abnormalities of carbohydrate digestion. Hope you have got a good idea about it. Thank you everyone for the patience hearing. Assalamu alaikum.